So, ladies and gentlemen, my name is Sharon Whiteman, and I'm part of the executive team at the Lyme Disease Association of Australia. And every year in May, we attempt to reach some of the brightest minds in tick-borne diseases around the world. And we're very privileged to have Dr. Elam. Now, she's the president and CEO of Galaxy Diagnostics. She co-founded Galaxy Diagnostics in 2009 and continues to lead corporate strategy and business development activities. Dr. Elam is an entrepreneur and a scholar with an experience in life sciences and information technology markets. In addition to her leadership role at Galaxy Diagnostics, she holds a Diana Research Fellowship at Babson College, where she conducts sponsored research studies for the Center for Women's Entrepreneurial Leadership. Dr. Elam holds a doctorate degree in sociology from the University of North Carolina at Chapel Hill and has published extensively on the topic of business, startup, and growth in top journals and global research reports. Her doctoral research on entrepreneurship was recognized by the Academy of Management for Innovative Thought Leadership, and Galaxy Diagnostics represents Dr. Elam's first foray into life sciences industry. As a highly regulated and complex industry, the life sciences provide countless illustrations of the barriers to innovation in healthcare and medicine. The challenges for entrepreneurs and business leaders today include finding better ways to build flexible, efficient organizations and processes to facilitate market innovation. God, I'd love to have hours to talk about, how innovation is so required and long past due in healthcare. And I think, I was in healthcare as a clinical nurse specialist in critical care. And I left because I was seeing the gaps, and that's been highlighted in my journey with Lyme disease. It's at a massive level. So, Dr. Elam, thank you so much for being here. I was thinking that it'd be great to start at the beginning. How did you come to co-found Galaxy Diagnostics?
So that's a great question. I actually, completed my doctorate in 2006, and I did a couple of years of fellowship. And so when I started looking for a tenure track position, it was 2008 in the middle of a recession, and the university stopped hiring. So I had to find something else to do. And I ended up taking a part-time position teaching at North Carolina State University. And that's where, I subsequently met my co-founders at the College of Veterinary Medicine. And they told me about this fascinating world of emerging infectious disease where they work, a technology that they were developing to basically solve a very sticky problem that nobody was really paying attention to. And a quick way of saying that is stealth infections, right? Or low abundance infections. So infections that infect at very low levels, that hide in cells and tissue and evade the host immune response. So all forms of testing are limited, right? Whether you're looking for antibodies, you're looking for DNA, or looking for other protein markers that indicate the presence of the organism, it's very, very challenging.
It must be very interesting.
Well, I was fascinated by it, and they had technology, which is the Bartonella alpha proteogrowth medium. So they had a novel chemically modified liquid enrichment medium that they were using to culture Bartonella. And the reason why they developed it is because, Bartonella were only discovered around 1990 in HIV patients. A lot of people don't realize we've known about cat scratch disease and trench fever for a very long time, but we didn't know which organism caused those diseases until we had this enormous population of immunocompromised patients. We learned all sorts of things about infectious disease during the HIV epidemic. Bartonella was discovered as a key cause of culture negative endocarditis. So if you knew the heart was infected, but you couldn't grow anything up on a culture plate, then it's going to be Bartonella. It could also be Coxiella burnetii, another zoonotic vector-borne disease. And it also caused, Bartonella was also linked to neurologic infections, so encephalitis, neuroretinitis, and all sorts of different presentations, including tumor growth, right? So what you remember that HIV patients would present with these mysterious lesions, and nobody could really figure out what they were. Some were cancerous, like Kaposi's sarcoma, but not all were. And it turned out that there was this little bacteria nobody knew about, causing this tumor growth, very difficult to isolate and culture on a plate, so very difficult to study. And the veterinarians really took on Bartonella as a key area of research focus, because many cats are naturally infected and harbor the bacteria at very high levels, hence, cat scratch disease. And so veterinary researchers have really kind of led the charge. Initially, it wasn't called Bartonella. It was considered a rickettsia, so in the rickettsial family. And so, there was this evolution and naming conventions and characterization until they identified exactly what it was. And the first species of Bartonella discovered was actually in South America. But this is the first time they found 2 species in HIV patients in North America. And that's what has led to all of what we've learned about Bartonella since then. And of course, I think in a very practical way for listeners, I think, you understand that infections are life-threatening to people who have compromised immune systems. I mean, we all know that, right? If you have a suppressed immune system, you're vulnerable to all sorts of infections. What evolved out of this era and the discovery of Bartonella was, this is not an infection they picked up after they became immunocompromised. It's one they already had in their system, right? So what else are we harboring, right? That may be contributed, it may put us at risk when we're immunocompromised. And what are we harboring that might actually be contributing to disease processes that just doctors don't test for, that we don't know a lot about? And so that's really what drove a lot of the discovery work around Bartonella. How prevalent is it in healthy people, these bacteria in healthy people? How, what disease states are they really associated with? Cat scratch disease historically was considered, self-limiting. In other words, you'd have this reaction, persistent swollen lymph nodes, and they just resolve on their own after 2 or 3 months, kind of like mono. Bartonella infection, cat scratch disease, is actually a key differential for mono. A lot of people don't know that either. And so it's this really fascinating little world they introduced me to. And I'll say that they, in an ideal world, these researchers, Dr. Ed Breitschwert and Dr. Ricardo Maggi at North Carolina State University, really wanted to hand the technology off to a big company and say, here's a tool, please go use it to do this important work. And it's just not the way innovation happens, right?
No, they don't want to
So we
change,
can
do
talk
they?
more about that, but I'm going to pause.
No, that's very good.
So
And
they engaged me to help them figure it out. Yeah.
that's so awesome, because I don't think you can infiltrate the minds of people who are in status quo. I think it, like the innovators, like what I started with at the beginning of it, that's how we're going to move forward in these difficult areas. So I don't know if this is a humanly possible question to answer, but is there any way that you can sort of explain to us in layperson friendly terms the evolution of testing, how it started and how it's progressed, and the innovation that's happening now, and in particular with Galaxy?
Yeah, I'm going to start in a really practical way. COVID gave all of us a crash course in infectious disease. And we haven't all really thought in detail about why we were given the guidelines that we were given, but really simply, medical authorities in the US, CDC, FDA, came out in COVID and said no antibody testing, right? Only antigen testing and PCR testing to confirm current infection with the COVID virus. Well, the reason why is because after natural infection, you get antibodies and they persist for a while. After vaccination, you get antibodies and they can persist for a while, maybe 18 to 24 months even. And with bacteria, they can persist a heck of a lot longer. And so, you think about some of the vaccinations we get as kids and how that confers, immunity or for much of our lifetime, right? And so we're just provoking the immune system to recognize these invaders, right? And so antibody testing is what they use for disease surveillance because it is quick and cheap. It's the bottom line. That's what happened with Lyme disease. Is when Lyme disease was discovered, it was really in 1977 in Lyme, Connecticut. A lot of people say Limes. It's actually Lyme, no S, Lyme, Connecticut. And with an outbreak of juvenile rheumatoid arthritis, right, in a group of children, researchers at Yale and area hospitals really kind of led the early discovery around Borrelia burgdorferi was the organism. And it was difficult to detect in blood. And of course, it was 1977, and that's when really that humoral immune response, the antibody response, that's how a lot of infectious diseases were tested, again, quick and cheap. And so what I would say is that a lot of science went into creating what they call the standard two-tier testing methodology, which is now being displaced in the big labs by modified two-tier testing. And the idea there was that you would have an ELISA test. So you'd look for one protein associated with the bacteria and then confirm it. This is standard two-tier testing, and confirm it with a Western blot that would look for a variety of different proteins that when the test was first created was thought to be, they were all thought to be quite specific to the organism. It turns out a lot of them aren't specific to the organism, which means that you could have another infection that can cause bands to light up, IgM and IgG. And so there's a lot of back and forth with scientists to figure out exactly how many bands you count and which proteins matter and which don't. Some of the specific proteins were taken out of the formulary because they were hoping for a vaccine. And they figured the vaccine would go after, those specifics, band 31, band 34, for example, were the two bands taken out. And so I think that there's this whole science behind how that algorithm evolved. It was originally developed for surveillance. It became the clinical tool because direct detection didn't work. You couldn't grow the bacteria on a plate in any timely fashion and with any efficacy. And so it just prevailed for a long time. And even with the development of some very exciting DNA test techniques we have, so PCR is the name of one type of DNA testing, where we see next-gen sequencing now, right? We're super excited about the high specificity of next-gen sequencing, but all of these tests have exactly the same problem. If you take a specimen from a patient and you have a tube, we only take out a tiny little bit to test. So even if you're lucky enough to have a couple of organisms in the tube, what are the odds that you're actually going to detect them in that tiny bit you take out for the test? And the answer is it's really low. So unless you have a high abundance infection in blood, you're not, DNA testing has limited utility. And so, of course, with the Borrelia burgdorferi, it actually escapes the blood after a couple of weeks and hides in tissue, sometimes in cells. And so it can evade the antibody response, which limits the utility of antibody testing, and it's near impossible to detect in blood after a couple of weeks. Not true, by the way, for the relapsing fever Borrelia. So there are other Borrelia species, right, that can also light up Borrelia antibody testing. So that's the complexity. We can either look for indirect evidence of the host immune response, or we can look for direct evidence. And that's what DNA testing is. Antigen testing is looking for specific proteins, usually surface proteins on the organism, kind of like you think about COVID and the spike protein. We're looking for surface proteins that are on the Borrelia. And that's challenging because not all of the Borrelia is famous for cloaking devices. It can change the surface proteins on the bacteria, but it does throw those proteins off. And so so there is a potential for antigen testing. The challenge is, could you find those proteins in blood? Probably not. So I hope that it's a little bit of background around the messiness for Lyme disease testing. And specifically what we're doing at Galaxy is we're going after that low abundance problem. So we're using technologies that if there are a couple of organisms, you can grow them up using the proprietary growth medium that were developed by Dr. Breitschword and Dr. Maggi at North Carolina State University. That's where we were born. we were spun out of North Carolina State to commercialize that technology, which, by the way, grows a lot of things, right? So blood microbiome studies, it would be a good tool in. But it can only grow the bacteria, or organisms if they're there. And so that's the challenge, again, coming back to Borrelia burgdorferi, is it may not be there. And that's where we ended up connecting with researchers at George Mason University that developed a urine antigen test for Borrelia. And this kind of goes back to research again. We know that, in mouse studies of Lyme borreliosis, the bacteria tend to aggregate in the bladder and throw off those surface proteins. And we know this, researchers will actually, terminate a mouse and confirm infection in their models by testing the bladder. And so that's where this idea came out, like maybe urine antigen's possible, but the answer is the antigens are there in such low numbers, it's really unlikely you'll be able to confirm. And researchers around the world have been trying to run DNA in urine and it didn't work. And so they created these nanoparticles that capture and concentrate target proteins. Different formulations going after different targets, but their first assay they developed was to capture and concentrate the OSPA surface protein in urine to confirm the presence of the organism, in the host, in the bladder. And so we took on that assay, and we've been working with the particles for quite a while now. And we're excited about the technology and think it can be broadly applied to a lot of different organisms, which they've already published proof of concept with. And then we're also developing PCR in blood using the enrichment medium, but digital PCR, which also tackles that low abundance problem, but in a different way, right? Instead of running one PCR on one tiny aliquot, it basically takes that aliquot, breaks it down into 10 to 20,000 droplets, and runs a reaction in every droplet. And the reason why that's important is because it reduces the background noise of the host DNA. Looking for microbial DNA in a host sample is like looking for a needle in a haystack. All that hay gets in the way, right? Reduces sensitivity. And so that's what we're doing. We're working with these 3 technologies. It's all about, if you're lucky enough to capture some organism in a specimen, how can you, or target protein in a specimen, how can you increase the odds of confirming the presence of that organism in the sample collected? Is that, I hope that's lay enough.
Yeah, no,
Yeah.
that's great. It's helped me because that's something that I've read and been with my little brain issues that I've got post-infection or with infection, whatever the truth is. I have a hard time retaining it. And it is a really thing for Lyme patients to grasp the difference when looking at this diagnosis, because it's unique in healthcare, isn't it? They, especially in Australia, you're showing up at a doctor and they say, well, it could be this. And then they're given this plethora of options for testing. And it's like a haystack. It's like finding your way through a haystack to understand
And
it.
it's far more complex than that. But the way, when I'm talking to people, I basically say there's 2 types of testing, right? There's direct and indirect in infectious disease. And there are a lot of other types of techniques in direct detection and a lot of other techniques in indirect detection behind a lot of the different tests that are available and that doctors have to choose from and that patients have to consider these days. And it's really complicated. The challenge, again, is anything that's indirect has specificity concerns. There are big risks, not only of false negatives, right, which is the big quite a problem with antibody testing, but also false positives, right? And antibody testing has limited utility when you expect to find those antibodies. And so that's one of the big points of controversy, or one of the challenges that we have in post-treatment. So for patients who have are diagnosed with Lyme disease and they're treated and still symptomatic afterwards, doctors don't have tools right now to distinguish between people who may have persistent infection versus those who have some level of immune dysregulation or some other complication interfering, maybe contributing to those non-specific symptoms, right? A lot of the symptoms are not specific to one infection or even to infection at all.
And so, to be clear for Lyme patients, the tests that would be considered direct are?
Our DNA testing,
Yeah.
right? And we have lots of different types of, it's also called nucleic acid testing. And so PCR is the most common. Sometimes you see PCR will be described as qPCR, it stands for quantitative PCR, or RT-PCR, which is real-time PCR. They're the same thing. And then there are other technologies like next-gen sequencing, NGS, and that is, again, they're looking to very specifically identify sequences in a sample. But NGS can identify multiple organisms in a sample, right?
Yeah.
It can pick up lots of different DA fingerprints. And there are a number of other technologies that are, being applied in Lyme disease right now on the direct detection side of things, really in research still. And I won't get into too much complexity there, but I think antigen testing is another type of direct detection. So I would say there's DNA testing, there's antigen testing, and antigen is proteomics. That's the complex scientific word, looking for proteins that are specific to the organism.
Okay.
And we think of those like with COVID is, or a pregnancy test, right? That's a type of proteomics, a type of antigen testing. You think about your COVID test that you can do at home, saliva testing, another type of antigen testing. Yeah,
Okay.
very specific. But then indirect detection, there are lots of different types of antibody testing too. I mean, we're not going to make this easy for anyone, right?
That was
And
my
then
goal.
I know, right? And then there's another emerging category that's going to make it even messier for patients. And those are immune biomarker panels or immunoassays, especially multiplex immunoassays, where we're going after, lots of different, usually looking for some kind of a unique signature of, immune proteins. So not something specific to the organism, but just a pattern of immune response that might be indicative of a particular disease state. And that's going to be super complex, right? Because you could have a signature that is, researchers, scientists are praying and hoping they can find signatures that are specific to an individual organism. I think it's extremely unlikely unless they're using other targets that are specific to the organism. Because infections, I think the best way to describe this, and I think it made sense the first time I heard it, but our medical director, Dr. Bob Mosiani, explained to me one time. He said, here's a doctor's job is really challenging, right? A patient shows up in the clinic and you do a workup. You ask them about their symptoms. You collect some other background data, do a full medical history. And he said, and literally there's only so many ways the body can express, symptoms. So you have disease. So you only have a short list of symptoms, and the possible causes are like a mile long. So the doctor's job is to narrow the possible causes into a short list and kind of test those. Think Dr. House, kind of go through and try to figure out which one's the most likely. And that's really stuck with me over the years. Like you really see that. And you see it in the research too. It's really challenging. And that's where vector-borne disease in particular, the index of suspicion for doctors and for patients really should start with, what's your risk exposure, right? Do you spend a lot of time around animals? Have you had high vector exposure? Do you have an occupational hazard? Like, are you a veterinary worker or animal worker? Are you an outdoor recreationist? Do you spend a lot of time hanging out and tick areas? Are you a gardener? Could they be in your backyard? Do you picnic at the city park? Right? These are risk exposure. And so it really should start there. But doctors often don't ask those questions. Like, what do you do for a living? Do you like to picnic in the park? Are you a trail runner? And so that's where our patient education is really important. We need to
Or were your
we need to
parents, right?
bring those. Yeah. And far beyond things like, did you have a tick attachment, or, have you recently been bitten by an animal, or scratched by an animal? And so it's basically combining that risk profile with the symptoms, where you start to get a sense that someone may have been at risk for Lyme disease. Their symptoms are consistent with what's been reported, and they have a high likelihood of having acquired it through, lifestyle or occupational hazard.
And is there any difference in this information? Should it be congenitally or sexually transmitted?
Well, I think that is a really great question. For most tick-borne infections, we would say a vertical transmission, right, which is congenital or perinatal, has been suggested, has been documented in certain cases, certainly for Bartonella and for Borrelia. It's been documented. The question really is, how common is it? Because that is another thing that goes into the diagnostic consideration. I think the way doctors often phrase it, right, is you hear hoofbeats, why look for zebras if it's most likely a horse? And that's where, everything new is rare until we know better. And so we think, it's a lot more common than maybe currently appreciated, but there's a lot of science that has to go into sort of proving that in a way that it would be included in continuing medical education, or convince medical experts who write the guidelines to actually mention it, kind of thing. In the meantime, patients are stuck, right? And I think sexual transmission is something very similar. If it is happening, is it very rare? What's the specific mechanism of transmission? I talk to patients about this all the time. Like, for example, with Bartonella has been documented in the saliva of dogs, cats, and people. So could you transmit it by kissing? Well, Bartonella needs access to the bloodstream. And our GI tract is literally designed so that organisms aren't entering the bloodstream. And so, but we know under some conditions that changes, right? So you think about leaky gut, you think about the consequences of stress or ulcers, or, sores in the mouth, or, other injuries that can actually now put you at risk for anything that you can swallow could also find its way into your bloodstream. And I'll tell you, the microbiome research is supporting that. Blood is not sterile, even in healthy people, and in sick people, blood has a lot of living organisms in it. And so we're really learning that whether they're leaking in from the stomach, whether they're coming in through the oral microbiome, or otherwise finding ways into the blood, that is sort of pathology research that scientists are trying to understand better.
And it's so crucial and underpins, I guess, pathways in healthcare going forward, because this is, like you said, these stealth and emerging infectious diseases are typically being ignored and overlooked in the medical field anyway, and health policy.
Well, and you can understand why ticks then are particularly evil little vectors too, right? Because they can inject the organisms right into the blood. Fleas are a little more complicated. Fleas aren't high on the radar for disease transmission. But they don't, for Bartonella, for example, fleas aren't injecting it into you when they bite you. They literally, like babies, they feed on your blood and then they poop, and then you itch and then you scratch it in, right? And same with the animal bites and scratches. It's literally, this is the high probability. It's possible that it is transmitted other ways, but the consideration there is that Bartonella transmission in the context of cats rarely happens outside of flea infestations, because the cats are scratching, they're collecting the flea feces underneath their nails, and then they're cleaning themselves, so it's in their mouth. And then you can think of that in a dosing way, right? It's in concentrations in the feces and the mouth of a cat cleaning those feces. And then if you see scratch or bite, all of a sudden you understand it's like the tick. You're injecting a high dose of the organism right into the host body. And that's, in those cases, we expect to see sort of a vigorous immune response. But it doesn't always happen that way.
Well, the reason I brought that up is because one of our colleagues at the LDA has a 30-year history of being sick, and after a tick bite in the UK, and but bullseye rash and a short fever, and then didn't think about it again and didn't connect that to a life of chronic disease at various levels. And recently, she, in part, decreased in her quality of life some COVID vaccines, and so made the decision that she really needed to go overseas for treatment, went to Germany. And while she was there, her son went as her carer. While she was there, they started having these discussions with the family and realizing all these symptom patterns in her 3 children. And her son was tested while he was there. His symptom groups, while chronic, and she's looking to my internet freeze, sorry. Admin announcement, my internet's dodgy in regional Queensland here. Yeah, so that they're looking at all 3 of her children now, to see, because she had 3 kids after that.
And that's really interesting, right? This goes back to this idea that it's very complex chronic disease. And you'll know this, that a lot of people being treated, doctors are measuring immune competence, and looking for even genetic risk factors for the body's ability to clear out infection, or it's very complicated. And so when we're looking at things like congenital transmission, it's really challenging. We think, it may be rare, for example, but environmental exposure is shared in a family. So in vector-borne diseases, this is like the hallmark, are cluster infections in families and in work groups. And so I don't know, you talk to a lot of doctors and they don't really care whether it's congenital or not, unless it's a pregnant mother and a baby. Because once the infection's there, the point is that whole family shared an environment, right? Where it does really matter, I think, are cases where the mother's very sick and there isn't a clear risk environment. Like they didn't spend a lot of time in places where there are ticks, but that's also really challenging too. And I think that this is something really important for people in Australia, where the ecological data doesn't support Lyme disease per se, although you do have ticks with other pathogens in them. Travel history. I can't tell you how many cases we've seen where an individual from a non-endemic area travels somewhere where Lyme disease is very prevalent, has tick contact, doesn't think anything of it, goes home, gets sick, goes to the doctor, and it's nowhere on the radar. So if you think about where people are most likely to be diagnosed with Lyme disease, that diagnosis is also more often considered, right? Because it's common and it's one of the things you want to rule in or rule out. But people who come from areas where it's a rare diagnosis, it's literally the bottom of the list. We're freezing again a little bit.
Yeah, apologies for that.
And
so did
you hear
the travel
all
history?
that? Travel history is incredibly important risk factor. And we also see some, some of the very unusual stories will come up through travel as well, like going to the Middle East and having sand flea exposure. And there are a number of organisms in those sand fleas. And that's where, finding a doctor who's experienced in travel medicine, or maybe getting a second opinion from someone who's experienced in travel medicine, may be helpful.
What about midges? They love me. They can get through all the screens in here and find me and at my desk.
I tell you, there are a lot of other vectors that we have a lot of questions around. And I'll, there's a lot of complexity for the patients out there if they wonder, why, doctors aren't taught to think about vector exposure more broadly, or, really big question, for example, here is Bartonella is considered tick-borne in Europe, but not in, not in North America. And but it's in all the ticks in North America. So a lot of doctors are like, well, clearly it's tick-borne, but the scientists are like, hold on. They proved it in Europe with artificial feeding models and mouse models. We haven't done that here with our ticks. And that's what they're waiting for. Even though some of our ticks share a very similar biology to the Ixodes ricinus Cape bean tick there. There's also a lot of splitting hairs that goes on around which ticks transmit which infections and at how high a rate. So we have a lot of ticks in North America who can transmit the pathogens, but they're not aggressive. They don't bite as often. It's possible, it's just not probable. And again, doctors typically don't care. They just want to know, was there a tick attachment, right? Patients don't really care about the specificity or the probabilities much either. They just want to know I was bit by a tick. Did it give it me Lyme disease or something? And the same thing goes with like which organism. So Burgdorferi transmitted by Ixodes scapularis is the deer tick in the United States, is the most likely way you will get it. That doesn't mean you can't get it other ways. In Europe, it's the Cape bean tick and it's other Borrelia species that cause Lyme disease, alphasic or NEI, and so on. And so I think there's this sort of geographic ecologies that contribute to definitions of what's endemic and how high their risk is. And, you know, hard for patients and frankly, a lot of doctors to navigate that.
In Australia, their ecological review sort of falls under the don't look, don't find category. They do have evidence that they're refusing to acknowledge. So how, like, it's all this, I think it's probably obvious answer to this question, but just how important is this innovation and scientific prioritization in the future for Lyme and co-infected patients, for diagnosis, for treatment? How important it is for the focus on improving and
well, if you don't have good
yes.
diagnostics, you can't develop good therapies, and from a research standpoint, we really desperately need direct detection, because we need it to be specific and real time, right? That we know the infection was in the patient. One of the most interesting things I learned coming into this that was unexpected, coming into emerging infectious disease, we're using the science to prove that there's a disease association. And what I didn't know is that labs define, false positives and false negatives differently from the way they do it in medicine. And so, for example, I had a conversation one time with a doctor where, he basically told his patient that he didn't believe the test result. He thought it was a false positive. Well, the patient, of course, came back to us with that and said, very upset, and said the doctor doesn't believe the test. I didn't get into it. I just called the doctor and said, because he was actually, one of our research partners. And I was like, talk to me about this. I know that we do good testing. So, but the patient thinks that you think our testing's terrible now. So like, talk to me about this. What did you mean when you told the patient you thought the test result was a false positive? And he said, what I meant is, I'm not sure. I believe the test. I'm just not sure if the infection is actually causing the patient's symptoms. I was like, okay. That's where you get into, that's a totally different line of scientific proof that's needed, right? It's, and that's really important when you think about organisms, for example, that may have, asymptomatic infection, right? Then how do you know what is causing the patient's collection of symptoms? If some people can live pretty comfortably with the infection, but maybe other people aren't, or is there something else going on? And so COVID really, with long haul COVID, has really kind of raised the bar on that whole level of questioning, has really started to change some minds about the role that infection can play in chronic illness, or infection associated chronic illness is how scientists are talking about it now.
Yeah, it's given Lyme patients a leg up, hasn't it? The focus on long haul COVID, so to speak. And I know people with long COVID, this won't be a popular statement, but people with long COVID, they're sick for a couple of weeks or a few months, and then the Lyme patients that it's been decades are going, oh my goodness, we wanted this kind of, but hopefully we'll get a secondary gain out of this research coming out of COVID.
I think so. And I will say, for long COVID, one of the things that we're seeing is that a good number of people, most people will get back to normal within a year, but some are remaining ill after a year. And that mirrors some of the data we have from studies that, follow lab confirmed cases of Lyme disease. So patients who have a lab confirmed Lyme disease follow them through treatment, but 40% are still symptomatic. This is based on research out of Johns Hopkins University, John Ocott's group there.
Yeah.
40% are still symptomatic at convalescent stage. And then by the end of the year, most of those people are better, but still there are 10 to 15% in his studies who are still experiencing significant disability related to that initial infection. And so that's long haul COVID and Lyme disease are very similar there. And that sparked a whole new line of research looking at latent infection and COVID long haul patients. Like maybe you had someone who did have a latent Lyme borreliosis and they got COVID, and that's why they're still sick a year later. Or, maybe it was Bartonella, or maybe there's some other infection. And that whole pattern we see with Lyme disease where like viruses like EBV get reactivated.
Yeah.
It's the same thing we see in chronic Lyme disease. It's the same thing we see in chronic Bartonellosis. It's just opening this whole new window and a whole new way of thinking about, the infectious causation or triggering of chronic illness.
So what kind of questions need to be answered for the future for Lyme and co-infected patients? You covered quite a few, but.
Right. Well, I think that that is a project that we're engaged in, right? So at Galaxy, we spun out of a university, our roots are in the science, but we're very open and interested in understanding what the doctors and the patients on the front lines are actually going through. And so I'll tell you some of again, I'm coming back to the low abundance problem, right? So the pathogens in the patient in low abundance, when you take samples out, it may be there in low abundance, right? We can tackle the lab piece of it, but we're working with doctors around, questions of provocation, for example. Is there anything the patient can do to increase, prior to sample collection, to increase the odds of getting it in a tube? And you'll hear people talking a lot of reports and a lot of speculation around things like, should I exercise first? Should I get a deep tissue massage? Should I do yoga? There are some pharmaceutical, challenges that different doctors are interested in studying. Like, is an antibiotic challenge a way to get a positive PCR test? Or what happens if you treat with steroids? Will that actually help you get a positive? These are, not something you want to do at home, but these are, you go ahead and do the exercise, massage, yoga, before you go get a blood draw. But I think these other techniques are something we definitely need to study under, controlled conditions, because we want to protect the safety of patients. So I would say there are questions like that. We have our team is also working on other collaborations around different therapies. And again, I think that's where direct detection is absolutely critical, right? We need to know what the prevalence rate is of the presence of the organism in different disease states. And we need to we need to confirm that. We need to understand what the disease mechanisms are. And we need really well-crafted research published in top journals that then can be included in continuing medical education for clinicians and healthcare providers more generally, so that they understand what the science is seeing. And Sharon, with your background in healthcare, you'll know we're spitting out knowledge faster than healthcare workers can keep up with,
That's
right?
It doesn't make it to the healthcare frontline.
Yeah. So I think we need direct detection to drive discovery. I think we need to understand the patient part of it, right? Like provocation studies. And I think with that, those advances on the diagnostic side, then I think we're going to be able to design really good studies to explore different therapies.
And then how to bring some of that innovation that's documented to be safe more quickly into the doctor's office, so that it would benefit patients, because it's decades now. It's 3 decades of stuff that's proven overseas. It's not made it to our doctor's office here in Australia. So that's a failure in policy.
That's an access challenge, right? And actually one that we've taken very seriously at Galaxy from the beginning. And I think everyone, listening will appreciate the innovation is really expensive. I think we all know that wealthy people get access to the new stuff long before the rest of us. And from an innovation standpoint, it's really challenging too, because we have to pay our bills, we have to pay our people, we have to pay, to move things forward. We got to pay for the studies, we got to do all this stuff. It's expensive, but we've done our best to really position what we do for mainstream, and we're partnering with large companies to hopefully get the advances that we're making into settings where it's run at high volume and the prices come down and there's insurance coverage, and international access will be facilitated under that approach as well. And that really does stand in contrast to like the other option we had, is staying small and being a boutique and continuing to work closely with the community. The problem there is you don't get the credibility around the assays. So we've chosen to invest in publication. We've chosen to put reimbursement codes in the US behind what we're doing. And we've chosen to, really do everything we can to position what we're doing so that it's really attractive to the big companies, and find ways to. So one of the things we want to do with our assays is we want to productize them. We want to create testing kits that we will sell to other labs around the world. So then labs in Australia could buy them and run the assays. So and every lab's coming at it, from a different perspective. But the only thing that solves the access problem is mainstreaming it, right? And that either takes 30 years, or you have to find, or you can get to scale faster through partnering and publication.
Yeah, that's awesome. So I was going to ask you what the hope is for current and future Australians sick with Lyme and associated infections. And that, you've just covered part of that. Is there anything else you'd like to add?
I can't tell you how important data is. So I don't know if you guys follow at all the work that LymeDisease.org is doing, has done with their patient registry, My Lyme Data.
Yeah.
I can't tell you how important that patient registry has ended up being. And I remember one presentation many years ago that Lorraine Johnson did, where she had a couple of maps on a slide where she said, this is where what the CDC says the patterns of Lyme disease are in the US, right? Concentrated in the Northeast and, dappled, not even at all in some states, right? And then this is what my Lyme data says. People live all over there's still a concentration in those areas, but people were reporting diagnoses of Lyme disease all over the country. And then she presented a third one where she was like, and they also surveyed the doctors, where doctors saying they're documenting the cases, and it matched the patient data, but not the CDC data. And what stood out to me when I saw this is that at Galaxy, we're a One Health company, right? So we're coming from animal medicine into human medicine. These are infections we share with our beloved pets that sleep in our homes and sometimes our beds. And we're very close, right? We do everything with our animals. And so it just jumped out at me because I'd been working on another project with the Companion Animal Parasite Council in the US. They have an interactive mapping project where they were using diagnostic data. So again, antibody surveillance data from a leading diagnostic company in animal health called IDEXX. IDEXX had licensed their dogs in the US are screened routinely with a test called the SNAP4Dx that looks for heartworm, but also Lyme disease, C6 peptide, and and a couple of other infections. And so they gave that screening data to the Companion Animal Parasite Council, and they were looking to see if they could use the canine data as measures of risk prediction for humans. And they have this data down to the county level in the US, so across 50 states down to the county level. And it was crazy because which maps do you think that data matched?
The Lyme maps.
Not the CDC data, which was confirmed cases, right? And so that's a long way of saying data is really, really important. Comparative medicine with animal health is really, really important. And if dogs are being diagnosed, then people are also at risk. And if you have an opportunity to participate in a patient registry, take it seriously, right? Like that day, your story is like voting. Your vote counts. And so getting your story out there, getting your case data, it might be a lot of work, right? Especially if they're looking for you to report on symptoms and supplements and everything else that you're doing. But that's amazing. What LymeDisease.org did with the MyLine data really changed the conversation in an important way, and is also generating data I think is very useful to patients. So for example, there are some patients who spent tens of thousands of dollars on therapies like stem cell therapy. Do you know that that is the single least effective therapy reported by Lyme disease patients?
Okay. No, I didn't know.
Right? So the patients are very adventurous. They're desperate to get well. They're willing to try lots of things. Well, share your experience and do it through a formal structure like that, because it'll be counted. And then you as a patient can go and you get to see how, what's working for other people. Is anything working at all? And so I think, again, being able to quantify people's experiences and really get a sense of what those patterns are really important. I know that in the US, ILADS has a patient registry project in development. I'm sure there's some, I'm sure in Europe, maybe in Australia, there are some other opportunities to do that. If there isn't in Australia, that's something that maybe you can find a sympathetic researcher and set that up like they did. LymeDisease.org, I know that they would love to share their experience and guidance on that point as well.
I did ask them when it was launched, but it wasn't possible for them that time. So maybe it's time to revisit that. And just for the listeners today, we did do a survey in 2012 and 2014, and that was published in, it's on our website, and it's also was published in our submission to the Senate inquiry in 2016. And that little map down there with the, I might go this way, that's the actual tick bite reporting map in Australia, but it was only 2 years data, that amount, in from about 2014 to 16, I think, and then our software failed, and we're fully volunteer run sick people organization, so we haven't reinstated, but we're working on the tick bite mapping to come back up as we speak. We needed ArcGIS volunteer. And
yeah, that is fantastic. What does the funding situation look like there?
None. It's not here in Australia, no.
Yeah.
And false positive is the go-to for doctors in Australia at every level, specialists, health policymakers, GPs. We have hardly any left, and most of them are deregistered or restricted or penalized. And only an infectious disease doctor can prescribe antibiotics for tick bite, which is a 6-week to 9-month wait period in Australia. So it's a dedication to chronic illness, really.
Yeah, and I'll tell you too, something for everyone to keep in mind. We learn about know a lot more about Lyme disease in terms of basic research than we have some of these other infections, but and we definitely have a focus on tick-borne disease versus infections that begin transmitted by other vectors or other means. But there are a handful of infections that have been implicated in human disease and in chronic illness specifically. In 2003, the National Academy of Sciences in the US, at the time they were called the Institute of Medicine, but they held a forum on microbial threats looking at the infectious etiology or causation of chronic disease. And that list of pathogens, I mean, the research is progressing. And so last year in June, NAS held another forum on infection-associated chronic illness. It's really remarkable to do sort of that comparative medicine and look at which infections are being implicated. Lots of syndromes, like you think MCAS and POTS and metabolic dysfunction, just all sorts of different syndromes or dysfunctions and endocrine hormone disruptions, adrenal fatigue. All of these things are reported in with all of these other examples of infection-associated chronic illness, like outside of tick-borne disease. So if someone says maybe you have Lyme disease, don't get too fixated on that. I mean, obviously, if you had tick exposure and attachment, but there's a larger infection hypothesis that's kind of prevailing that says, could you be harboring a pathogen load? So there's a concept called the pathobiome that's really kind of developing in the world of tick-borne disease, for example, but also more broadly around, how infection may be contributing to chronic illness. Like, what do we accumulate over our lifespan that may put us at risk? And I think for people who suffer from chronic UTIs or chronic sinus infections, like, they totally get that. You often hear those people that get treated for something else with antibiotics, and then they don't have UTIs for 2 years, or they haven't had a sinus infection in 5 years. You get people who have bleeding ulcers, so Helicobacter, they're colonized with Helicobacter pylori. C. diff. There are a number of different organisms we can kind of collect over our lifetime that put us at risk for different illnesses. I think we froze again. We just lost Sharon. Okay, great. But I was talking about the NAS conferences.
Yeah. So
Workshops.
apologies for that internet outage. So we stopped midstream, and then I took ages to come back in. So thank you for your patience, Dr. Elam. It's not usually this bad, but anyway, we persist. It's just like our journey. We persist.
Yeah.
So you were talking about the importance of data and what they're finding and how that's matching up with the same picture for Lyme patients, and inviting people to participate in data collection.
Right. And then I also shared some of the broader research looking at the association of infection with chronic illness, and described some of the workshops that were held by the National Academy of Science in the US, one in 2003, and then again last year in 2023. And the infections are the same, right? So, but we have 20 years more data. I've historically, when I've talked about this as a new frontier of medicine, the role of infection in chronic illness, right? Microbiologists call it the new germ theory of disease. It's associated with, concepts like the pathobiome.
Yeah.
And it's a very exciting new area of medicine. But there are a lot of infections that seem to be linked to this. So I think that the takeaway for people in the world of, to get drawn into the world of tick-borne disease is just understand the disease complexity and know that, and doctors aren't well trained at this. So finding a specialist is kind of important, and it's very challenging because there aren't very many in the world right now. But disease is complex. The patients that I see recover the best are the ones that their clinicians don't just treat infection, right? They also treat immune dysregulation or hormonal disruption or whatever your complicating factors are.
Multi-system.
Yeah, it is really multi-system, and I have seen a lot of recovery stories. I think that's the other thing I always want patients to know is we have some of our patients have started a number of different patient forums, which I'm invited to comment on research or offer up studies or comment on technical things from time to time. But it's really challenging because when people get well, they leave. And so when you show up at these forums, the only stories you hear are the ones where people are still struggling.
Yeah.
And every now and then, we have some fantastic moderators in these groups, and every now and then, they'll reconnect with a patient who's doing great and say, do you mind just coming back in and sharing how well things are going for you? Because I think people could really use a little hope.
Absolutely.
And so
what what is in your heart for hope for Lyme patients in Australia to complete with?
I think it's the same for patients everywhere. I just want you to know there are an army of scientists working on these problems, that we went from no research funding to actually starting to see research funding. And the knowledge is moving forward really quickly. There are people like, me and others on my team who are really fighting the commercialization game. So the same way where 15, 20 years ago, there was no research funding. Now the trick is there isn't much commercial funding. So we're having to convince investors, right, that this is a real market, that people are going to pay for testing, that this is a good investment opportunity. It certainly is the right thing to do. We know that, but to translate things from the lab and into clinical practice, it really is, it takes a village. And we're working really closely with the regulatory agencies. Civil discourse, you guys, we really need to work together on this. I know it's frustrating. I know you want to, I know we all have those moments where we want to rage at the world, but also understand that there are a lot of good people trying to bring about solutions here. And just remember that it's an emerging infectious disease. So the hope is that people are getting better. The community of doctors who are having a lot of success with patients. No doctor has full success with all of their patients, right? But we're starting to grow a community of highly experienced clinicians who are having more and more success every year. The scientists, what's going to convince them that these infections are worthwhile are the severe cases. So you might feel dismissed or downplayed if you're presenting with something like chronic fatigue or fibromyalgia or something else that's MCAS or POTS or something that's sort of ambiguous, but something that you should be able to live with. I can't tell you how many patients have been told that, right? You're not sick enough to do anything, to do any big interventions. I think that community of physicians tackling that is growing. In the US, we call it functional medicine. So emerging area where we're looking at a lot of different approaches to getting patients back to wellness before things get too severe. So yeah, that's what I want to know. I want them to know the research funding is there. Advocacy is more powerful than ever. It's on the federal radar now in the US. Our Department of Human Health Services federal office has got a public-private partnership with a private foundation. Cohen Foundation has brought in tens of millions of dollars to this. CDC is working hard to, change the approach and get the right experts behind. NIH funding, we're seeing higher authorizations than ever before. And I hope that, the advocacy community in Australia can, gain some hope from there and maybe work with some of our advocates like our Center for Lyme Action and figure out what you can do where you are
That's
to
awesome.
move things forward.
Yeah. And well, I'd like to thank you. And on behalf of Australian Lyme patients, we are starting our data projects up this year in the month of May, which when you'll be watching this video. And so we invite you to participate. So again, data is key. Be able to show what's really going on. And
absolutely,
the government was requested out of the Senate inquiry to do an epidemiological review of all the recommendations. That's one of two that they've refused to progress. So, it's time for us to link arms as affected persons, families, friends, communities, potentially affected people, and push some of these reporting and data collection opportunities that we'll be launching.
It's a global social movement. It really is.
Yeah.
And everyone's working together, trying to learn from each other and really push the needle forward. It is working. I just want everyone to know that it's working over here. It's going to work over there. It's a matter of time. But again, every voice counts, right?
Thank you very much, Dr. Elam. Appreciate your time and expertise and passion. I love innovation. I love being around it. Thank you.